Someday I will visit yours, please.
So I think what we can start is Dr. Hanif is already checked in earlier. He will join in a minute. And he's one neurosurgeon who is also coming, and he will join me here, and we can have some interaction with him. So I think, as you know, the once a neural injury occurs, whether it's in the brain or in the spinal cord or in the peripheral part of nervous system, it feels an entirely different way. There's gliosis there. The gliosis is a very different tissue. It's similar to what's fibrosis in the ordinary tissue, and I think that creates a very sort of difficult situation in a way that it is difficult for the reparative tissue to overcome the barrier of this gliosis, and so to have a regenerative process is kind of stopped there, and the more severe, more prolonged, more sort of length of injury is there, then the more the problem is there to in regeneration and restoration of the neurology. With the new thinking and with the new techniques, the stem cell have come up as a very promising arena in which you can use stem cells or the stem cell products, which will help in um, overcoming this gliotic issue and start neurogenesis or delivering a new sort of growth factors which will help in resorption and overcoming the impediment of reparative tissue, which is, as I said earlier, which is not very easy to jump across and cause reconnection of the neuronal tissue. So I think in that respect, we are very, very much honored that we have two eminent people sitting with us, Professor Bambang Darwono from Indonesia and Professor Kuldeep Sidhu from Australia. Both are, in a way, the Kuldeep is very sort of innovative and pioneer in the stem cell therapy and the stem cell product therapy. And the Darwono has the similar experience. On top of that, he had a clinical experience in treating such patients, which is, to my mind, it was very encouraging. And I was, in fact, very sort of excited when I went to see him last year. And we really were excited, and we thought that we should do something about it. And hopefully, today's meeting will help us a lot. Over to Bamba.
Yeah. Thank you. So, shall I share?
Kuldeep, you want to say something before Bang Bang comes? Oh, you're welcome, my friend. How are you?
Cool.
Hello, Dr. Hanif is here. Professor Hanif, he's the orthopedic surgeon in one of the university hospital. And Professor Desmond Kwok, he's from Canada, and he's with us, and I'm so excited to see him. And I like your color of your jacket.
Oh, thank you.
Yeah, you're looking so young.
To brighten my day because it's a bit cold talk in Canada right now.
You're looking very young and very tight. We have just started that we'll hold this meeting in this spinal and this cerebrovascular of the brain, hemorrhage of brain injury. And once you have this injury, you have a gliosis, you have a damage there, and the reparative tissue somehow it's difficult for the reparative tissue to come across that reparative the gliosis. And now I think the stem cells and their products, they are coming as promising agents to provoke repair and come over this neuronal repair in a much more effective. And I think the Professor Kuldeep Sidhu will introduce the subject, and then Bang Bang Darwono will request him to tell us about his experience, and then we'll discuss along with that.
All right, thanks, Dr. Tariq. I don't know what I'm going to say, but suffice to say that this field of stem cell biology is evolving quite tremendously worldwide, and people have jumped into it quite prematurely to do some clinical application of stem cells. So the major issue, I think, in this area comes from ethical point of view, number one. And the second point I want to make is the quality control. People are not very much worried about it. And then the control trial and the publication. So the whole field of stem cell biology is suffering because we have not taken into consideration all these 3 points which I have referred to. So one has to be very cognizant of the ethical approval. One should be very cognizant of the quality control. And the model which I've been proposing for the last 15 years, that this clinical translation using stem cell cannot succeed unless we have a collaboration between a clinician and a scientist together. So you have to have the team of scientists and a clinician together under one roof to make a difference. Unfortunately, scientists don't know some of the clinical aspect of it, as Dr. Tariq very well mentioned about it. And similarly, the clinicians sometimes cannot make huge assessment of what they are picking up from the patient and what they are transplanting back into the patient. So quality control comes into play at that stage, which is very, very critical role. And then I think we need to follow through a ethical approved control process of drug discovery and then publish that data. So if we follow these 3 steps together systematically, this field can do really wonder. Tariq has very well discussed that this is an evolving field and it has a lot of scope in future. From my personal perspective, my background have been in stem cell biology using human ESL. So we were the first one to have a license in Australia to produce the first human ESL line called Endeavour 1 and Endeavour 2. And those lines are sitting with NIH for wider distribution throughout the world. So that is one of the credit I have in Australia. This was followed up by a lot of controversy using human embryos for producing this human ESL line and using them for therapeutic purposes. This was followed. I'm talking about this was evolved in 1979 when Jimmy Thompson from Wisconsin were the first one to isolate the cell line. I was the first one to come and join his group. At that time, learned how to do it, and then set up my own in Australia and produce the cell line Endeavour 1 and Endeavour 2. But because of the controversy using human embryos, Shinya Yamanaka from Japan, as you know, was the guy who got the Nobel Prize in year 2012. He said that you can reprogram cell to the same extent as that of human ESL using somatic cell by reprogramming. So that was a dramatic change in the field. Now everyone has been using, they are called the induced pluripotent stem cell, and these cells have been used extensively, not only for diagnostic purposes, but also some of the therapy with the eye treatment and even the spinal cord injury. People have been trying to using iPS-derived neuron, the spinal neuron, with this particular technology. So we had an international consortium, which is called ICSI, which is a quality control of stem cell worldwide. So one of my lab and 52 different labs in the world, they were involved to work on the quality control. Shinya Yamanaka, who's a Nobel laureate, we worked together. And I think a couple of years, we published a paper in Nature, which was published on quality control. So having said that, that's kind of my background in terms of using pluripotent stem cell, which has a huge potential. We can produce all 220 different types of cell in your body and can use either for diagnostic purposes or for therapeutic purposes. But we have to develop this field to an extent that it is more acceptable to the regulatory framework. It is it is accepted by the ethical committee that this is doing the job, what we are claiming that it is doing the job. then probably at that stage we are in business. So about 5 years ago, I'm still holding a chair of stem cell biology in University of New South Wales Faculty of Medicine position. But then I moved out 5 years ago, set up a company called CK Cell Technology. I can present to you as one of the video at the end of the conference. Um, the primary aim was to use adult stem cells. So this might be a commonality between all of us here for this particular meeting, which are evolving much faster than the purebred stem cell. So the adult stem cell we are specialized on are the mesenchymal stem cell, the American called mesenchymal or mesenchymal stem cell. So my lab is specialized in synthesizing, in a way, or manufacture these mesenchymal stem cells derived either from fat tissue or from the dental tissue. And we have the clinicians and technicians all working together under one roof. We have a GMP facility where we generate these stem cells, and then at the moment we are using those stem cells which we have produced. We have got quite a few number of cell line derived from a normal patient and also from the patient, maybe an Alzheimer patient, which is very close to my heart, which I've been doing for a long time, and then number of other ailments. But at the moment, I think focus is to have a quality control on this cell line. So we have produced serum-free, xeno-free cell line, and then I mentioned the ethical issues and the concern about using stem cell has been huge worldwide. It's still there, and I respect people who have a concern with ethical issues. Having said that, I thought that the focus of our company would be more on stem cell derived products. So that is, I think, the focus of discussion today, that the product derived from the stem cell like conditioned media and exosome could play a very significant role. And particularly, this role has become much more important with COVID-19, which is a very, very peculiar cytochrome storm coming up with the viral infection in these patients. And these mesenchymal stem cell and the conditioned media and the exosome can play a very significant role. And I'm really honored to associate with Professor Tariq, that we are collaborating together to do some pilot studies using our stem cell derived product from mesenchymal stem cell for spinal cord injury and also for diabetic foot ulcer. And we're also trying if we can do some trial with the COVID-19. So this is the kind of background, and I'm really excited to meet you all and learn from your experience and look forward to more of collaboration. Thank you. Over to you, Tariq.
Thank you very much, Kuldeep. I think very interesting overview and the basic sort of understanding and the conceptual background behind all this regenerative arena. I think you have explained very well. May I welcome Professor Hanif? Welcome. Nika, you're welcome.
Internet, some connection issues.
It's okay now. Can you hear me?
Yes, I can.
Yeah.
But your video is blocked.
Now, may I request Professor Bambang to say something?
I think there is some issue with the connectivity.
Hello?
Yes.
Yeah, we can hear you.
Yeah, we
Oh,
can hear.
okay.
Yeah.
Yeah.
You can
So,
hear me?
Yeah, we can hear you and we can see you all.
Okay. Okay, Bambang.
Yeah, I only have a small experience using the exosome. uh, why we used exosome? Because if we know that about stem cell, if we want to introduce to the human body, we should multiply, multiplicate them until 100,000 and then give to the body. And reported that only less than 1% is survive because they have antigen at the membrane. Now, in our team, this is research in this country, we then isolate the stem cell in the media without antigen and make them in stress condition like hypoxia. Then what happened? This cell is trying to keep survive and produce a lot of protein and growth factor, what we call it secretome. Then we collect all this and use as a treatment. So our treatment will be a cell-free therapy. And I have only just a few small experience with the patient, and I will show you later. Now we move again to the exosome, because exosome is a family of the secretome also, but this is small vesicle, but it contains a lot of protein and a lot of the micro RNA and everything. Then this is actually used as a cell-to-cell communication, and they can be transferred. And what interesting is, in infection, they can hack this biogenesis of the exosome and put their genome in that and become exosome infectious things and spread to another cell. But because this is the domain of the exosome, so if we give a normal exosome, healthy exosome, they can do a gene silencing through the microRNA. So this is very interesting. Hopefully, it will become the future of the treatment.
Nisha, would you like to say something?
Thank you very much. We all know this spinal cord injury is a devastating illness, and it not leads to physical disability, but a lot of psychosocial aspects. So, do you listen to me?
Yeah.
Okay, thank you. So, we see the patients of the spinal cord injury, the misery of these patients. We see a lot of such patients in our society, and they are suffering a lot of misery here in our society. So something we were in were in search of something to take out so that to reduce the misery of these spinal cord injury patients. So either we are over enthusiastic or maybe something is good for these patients.
I think you're very right, Hanif, that it's not only a physical issue. Physical issue is one aspect, but along with that, there's a huge burden of psychosocial element as well, because most of the patients, they belong to uh, constrained socioeconomic level, and once they are rendered incapacitated because of injury, it becomes difficult for the friends and family to look after them, and their participation also is limited, and they develop kind of sense of uselessness, which I think the whole idea of treating this patient, or considering the idea of treating the spinal cord injury with this new sort of technology or ideas, is to try to restore some function and some ability, so that they can improve from the scale one where they had after injury, where their neurology is not recovering anymore, and then you give some stimulus to the tissue to grow itself and regenerate and restore the functional ability or the sensorium to a certain extent, so that they can activate their certain group of muscle or the certain part of muscle, and then they become sort of better able to combat their physical or psychological impairment. So I think in this respect, this new technology or the new field of regenerative medicine carries a lot of promise. And I think Kuldeep is very right in saying that these new things and the Darwono's experience these things, it is very encouraging. Bang Bang, your point of view?
Oh, okay.
Yeah.
Shall I start? Thank
Yeah,
you.
please.
So this is my presentation. We remember this picture 1 year ago in Jakarta.
Very, very interesting picture, very historical.
And now, what are exosomes? This is small vesicles, 30 until 100 nanometer, associated by many cells, including the immune cell, cardiovascular, and tumor cell, and released to the extracellular environment, and exhibit a lipid bilayer with a marker protein and also cell type specific markers. I cannot.
Okay.
Yes, and contain protein and nucleic acid as messenger and microRNA, and play the role of cell-in-cell communication, and have a self-free regenerative potential, and naturally found in body fluids, including the cerebrospinal fluid. So in the biogenesis consists of 4 stages: initiation, endocytosis, multivesicular bodies formation, and then secretion. They also during biogenesis sorting the protein, lipids, and RNA. So what is content inside exosome? That is a lot of proteins, lipids, and nucleic acid, the messenger and microRNA, and also long non-coding RNA. So this is the protein composition in exosome, very complex. Oops. And this is the Myrna packaging into exosome. Then we can see the Myrna profile in exosome, also very complex. So exosome actually is a cargo, nano-sized cargo. So as a cargo vehicle, what interesting is it can penetrate the blood-brain barrier. That's why I inject the exosome into the LCS, yes, intrathecal. And maybe in the future, we can use this vehicle to use as to carry drug for other treatment. So how does it work? Uptake exosome by other cells, including the neuron cell. First, intracellular signaling initiated, then RNA protein transferred, and then at last the entire exosome transferred. So what do exosome do? Antigen presentation, dendritic cell, immune regulation, and also spread of infectious disease if this exosome is already infected. Epigenetic changes by Myrna promoters, because Myrna can go manage the gene and then change the gene expression. Now we are talking about the tissue repair regeneration. So here is how exosome can do a lot of things. We will talk tomorrow in coffee, but now we are talking about the regeneration. So this is the central dogma in neuroscience by Santiago Ramon Agaya. Once neuron development was ended, in the adult center, the nerve parts are something fixed and immutable. And everything may be die and nothing may be regenerative. This is a dogma for a long time. But then, this dogma is broken by a report of Gates and Temple. that adult neural cell system and neurogenesis were discovered in 1960. So this neural progenitor cell that gave rise to many, if not all, the cell types, neuronal cell types. And this is found in the two zones of dentate gyrus in the hippocampus brain, as well in the striatum part of basal ganglia, neurocortex, outer thickness of convoluted cerebral cortex, and also in the spinal cord. So neurogenesis is the ability of neuron to regenerate through the neuroprogenitor stem cell. In spinal cord injury, the key point is this is biphasic. Primary injury will lead to a cascade of deleterious effect in secondary injury. It means that early intervention will promising a therapeutic opportunity. And we should know the understanding of potentially beneficial endogenous response. It will be the novel therapeutic interventions. This is endogenous response, astrogliosis. Reactive astrogliosis, abnormal increase in the number due to the destruction of the nearby neurons by trauma, infection, ischemia, stroke, immune response, neurodegenerative disease, including Alzheimer and also Parkinson. And reactive astrogliosis occur prominently in response to all forms of the CNS injury or disease. And this is also endogenous of the microglia as a phagocytosis cell and oligodendrocyte to support insulation by creating the myelin sheath. In spinal cord injury in chronic phase, more than 6 months, volume degeneration occur and debilitating and irritating pain can develop. And what left is the multilocular cystic cavity in the spinal cord. And within 1 or 2 years, the lesion has ceased to progress and continuing disease are stabilized. This is the dogma of Raymond Ekaya. Right, because difficult to retain because there is a glial fibrous scar. At the beginning, it want to protect the injury from the primary injury to secondary injury. But at the end, this also prevent the recovery of the neuron itself. It depends how much it's destroyed, injury. So this is brain injury. Brain ischemia result in the upregulation of the endogenous neuroprogenitor cell. Now therapeutic approach is focused in augmenting the brain normal endogenous reaction to injury and induce neurogenesis by triggered by numerous neurotropic growth factor, and this is all available in the exosome. Also, increasing endogenous by non-coding RNA hormone, and delivery of this will reduce NPC depth, and it becomes neuroprotective effect in for stroke recovery, and also various chemokine receptors, stromal derived factor 1 and integrin beta 1, beta 1. This is all available in the exosome. So now I present a case report. It's a small case, 4 cases for spinal cord injury and 2 cases stroke. This is the first, 48 years old, burst fracture T7 with complete spinal cord injury, paraplegic Asia A, 9 months after injury and fusion, and now we can see in chronic phase a multilocular cystic cavity. This is 0, all the muscle chart, both leg, and cannot move. Then we inject, at the first time is using secretum, intrathecal intrapain. And we know later, 1 year ago, we start with exosome. And this is the result. In 3 weeks, he start to move the muscle belly. In 6 weeks, he start to walk, even still passive, using parapodium. Sorry. Yeah. And then he start to walk with walker, but not so. And after exosome injection, his hip and also the femoral muscle getting stronger. And he told me that this is the first time I can see my house in my own feet. And we can see the size of the cyst getting smaller. Also here, the improvement of the hip muscle and knee muscle, or 0. Second case, male 69, tetraparesia SCC, bedridden, neck and back pain, convulsion, after cervical surgery 4 months after in another hospital. Hope you know this also. And but he also has multiple lumbar canal stenosis. So we perform PSLD 4 levels and then inject secretome intrathecal, and we remove the cervical implant. And we can see here this is before. Then we do PSLD, and this is 5 days. Still very difficult to stand. It should be assisted. Also, if stand not long, he should sit again. But now he start to use the right hand for eating and start swimming. Then he's after exosome injection. He stand on his own, and now he can walk with very stronger the leg. And look, he can wake up by himself. So, number 3 is stroke. Male 40 years, 1.5 years ago stroke, non-hemorrhagic and operated craniotomy. Is still in wheelchair. Is after 1.5 years, right hemiplegic, motoric aphasia. This is the MRI before treatment. Then we give injection 4 times, interval 1 month with secretome. Then we can see the improvement of the MRI after 4 months. And he came to my practice walking with 1 cane. And better communication, no convulsion, no tremor. Then we use injection with exosome. Then a fantastic result after exosome injection. He start to tiptoe. And also he can do treadmill. So case 4 is male 22nd year, 292, military exercise accident, spinal cord injury, and already operated 1 year ago. He still has a problem with erection dysfunction, retention urine, alpha, pain, parasia, hypatia, motoric SSC. So we give the first injection, exosome. This is the already operated at the lumbar. There's another problem in the T12, but not operated. And this is condition before treatment. He should walk with 2 canes. Then we give injection. So this just 3 weeks after injection, intrathecal exosome. He can walk with 1 cane. And 4 weeks later, he can drive a motorcycle. Five weeks later, he can do a jogging. This is younger and SSC. Now the case 5 is 90 years with spastic tetraplegic, bedridden, pen sitting and moving leg. 3 years ago got a motor accident, cervical injury 3, 4, 5, 6, and already operated with laminoplasty 3, 4, 5, 6 in another hospital. He came in this condition, spastic hand, cannot sit, it was very painful, and we can see a lot of multiple cysts in the cervical. And this is condition before, very spastic hand. but he doesn't give up, still learning with cellular phone, try to learn something. He cannot go to school. And also his leg is spastic, cannot see. So then we give injection. Now just 4 days after exosome injection, spasticity decrease a lot, and he can sit and happy face. Just the first injection. And this is the sixth case. 77 years. 8 years ago, non-hemorrhagic stroke, right hemiplegic. Already treated with DSA 3 times, no result. 5 years ago, intravenous stem cell 28 times, no result, only the skin getting younger. 3 years ago, craniotomy and put stem cell into the brain, still no result. Then we gave injection intrathecal with exosome. This is the right hand and the right foot. Only move like this. Also the right leg compared to the left leg, and we injection. Just 1 day, he start to walk. It's very fantastic. Now, after clinical investigation, we start with basic science research. So we use Sprague-Dawley white male rat 33. We make artificial complete spinal cord injury. We can see here the, sorry. What happened? Sorry. The hind foot cannot move. And then 7 days after, starting to move the hip. And 2 weeks later, the foot is stronger and the rat can run again. And we make also histopathology. This is after injury. This is 2 weeks later. The astrocyte in white matter substance. This is in gray matter. And 2 weeks later. And also here, the spinal cord. 1 week later, and 2 weeks later, the normal spinal cord. So second treatment rat healed faster compared to the control in an incomplete spinal cord injury, acute, subacute, and intermediate. So now to conclude, the effect of secretome in case report improves the natural healing of 2 cases with spinal cord injury, chronic ASIA A, intermediate ASIA C, and also in chronic brain injury in case 3. In animal study, we can see regenerated neuron cell in rat with incomplete spinal cord injury, and exosome improved the natural healing faster compared to secretome in 6 cases. Our first animal study, we cannot create a chronic phase of conflict because rat already died before it's achieved. And we use now in ongoing still with dog that we make complete spinal cord injury and treat with secretome to compare with exosome treatment. So this is our team research in Dermama Biotechnology Laboratory, and this is our laboratory, and this is the staff of the proteomic, and we can make peptide profiling with the proteomic bottom-up system laboratory. Thank you very much.
Yeah. Thank you very much, Bambang. Very exciting. Tell me how, just within 24 hours, you expect or experience such a remarkable recovery that the patient is up and about. And have you ever done repeat MRI subsequently once patient, once who was riding motorbike you done the repeat MRI, that how much repair has been carried out at the site of previous injury?
Yeah, we only can do the indirect follow-up with physiotherapists. We can check also the muscle strength before and after, and we can make also MRI. But sometimes MRI not so easy to see the changes. Yes, but clinical is very important. And why we start that? Because the patient is come to me already go everywhere and fail. One under 8 years also. So when patient come to you with like this, do you keep do nothing or try to do something? Yes. So there's because the exosome has a potential treatment, then we give them this treatment, and I told them with that if we look that this is getting better, then I will give it again. And it better and stop. Maybe that is the natural healing of the patient is stop only then. Because who heal is the natural healing of the patient, not the doctors. We just to show the way and do something to help.
Desmond, do you have any question or comments? Come on, fire all the guns from the Canada.
Hello, can people hear me?
Yes, we can hear you.
Okay. First of all, it was very impressive presentation. Thank you, sir. I was wondering, you had done a comparison between stem cell and also exosome. And with stem cells, you did not get quite as robust results as you got with the exosomes. And exosomes are hard. First of all, we would like to know, okay, what type of stem cells were used, and what would be the scientific justification that the parent cell would not produce as robust of a response as the exosome was could?
Oh, okay, good question. Because exosome we also derive from stem cell, but we use the mesenchymal stem cell because mesenchymal stem cell produce a big exosome compared to another stem cell. The second, if you use stem cell is always identified in the cell membrane as an antigen by our body, and then is attacked. So from maybe 100,000 stem cells we put, only less than 1% is alive. And 1% stem cell is actually stupid, doesn't know what to be. Some become the expected cell, some become not expected cell. And the second thing is stem cell always carry DNA. If the DNA will become a tumor, it grows a tumor. It already reported. Now we try that what is the challenging hope of the success of less 1%? So we think about secretome, because the stem cell also working with that kind of communication to the other cell. So we now do in laboratory to excrete do a segregation of the protein, and we use this as a treatment, cell-free therapy. Then in 1 year later, we try to find what is the more of the active of this family group of vesicle. So we find this is the exosome, the smallest part, and this can also pass the blood-brain barrier, and it contains also a lot of protein and nucleic acid. So this is why I think exosome is also derived from stem cell, but maybe 2 steps ahead after that.
Okay. Now, the stem cells you used were also the injections you made were also intrathecal. So the blood-brain barrier should not really matter for exosomes, because you're injecting intrathecal from what I could tell from the pictures. Is that correct?
Yeah.
So blood-brain barrier would not be relevant here. Now, the other thing is, so you said that 1% of the stem cells survive. So even if 1% of the stem cells survive, that still would be hundreds of thousands. Do we know if the surviving stem cells, do they produce exosomes or not? And if they are there, continuously producing exosomes, won't that be a more robust supply of continuous supply of exosomes? I mean, that was my question.
Yeah, I don't think that the stem cell treatment is bad. I will say all treatment is good, but the best is how to use them. So I'm thinking about this exosome is what also stem cell is doing to have another cell. Then we just concentrate on exosome to treat the same patient.
Okay, thank you. Also, I just wanted to say hi to Dr. Sidhu, and I just wanted to tell him that I also got 2 degrees from University of Wisconsin, including one PhD, and I was working in the same department as Jenny Thompson, which was anatomy department in medical school. So it's always very nice to meet someone who has also gone to UW Madison.
Excellent. I really appreciate that, and that's a fraternity from Wisconsin.
Yes.
But can I ask one question to Bambang? What your preparations were autologous or allogeneic? Were they derived from the same patient and then injected back into the patient, or they derived from somewhere else and injected into the patient?
This is allogeneic. We can use from the fat, from the umbilical cord, or anything. But this is not cell.
And my follow-up question is, do you have a biobank of these exosomes, or you prepare at the same time when you are treating the patient?
Yeah, we have the bank, but the crucial is how to storage, I think, yes, because Dr. Hanif already has experience. When he bring this and then there's a problem of the flight and the storage maybe not proper, maybe, I don't know. And then the exosome is not working as well.
So how do you address that question in your lab, in your clinic?
Yeah, I put in the refrigerator in the refrigerator, just refrigerator. Then before that, I remove and put in the normal temperature in 5 minutes, and then I inject to the patient. After dilute with NaCl, become a 5 cc NaCl, then we inject them.
Yeah, but I think from our experience in our lab, the shelf life of these exosomes, including the growth factors, cytokines in the conditioned media, is very limited. So they don't have any shelf life whatsoever. So when you're injecting even after 2 hours, it doesn't make any sense to me, like in terms of the potency of those, which is going back into the patient. I think somebody has to look at how to maintain the potency of the cytokine, which I think is very dear to us. We put a patent last year how to preserve the shelf life of the conditioned media and the exosome. The trial we are doing at the moment is based on developing an appropriate technology whereby we increase the shelf life of these biomolecules before drying onto the patient. So that's my one question, but the other relevant question goes back to what Dr. Tariq said, that getting a response in 24 hours is something it's very difficult to convince the scientific community, whether it's a placebo effect or something else. We need to be very careful of and also publish our data so that people know what we are talking about and we understand the mechanism. Otherwise, I can see beautiful work you are carrying out in your clinic and the patients are getting better. These are the individual cases, but we need to do a proper trial, develop a kind of a approach where we do the clinical trial with the placebo effect and having a control trial, and then publish it and convince people that this is how it works. I know it works, but we need to convince the scientific community before we take it to the market and help those patients.
Thank you.
Thank you, Dr. Tariq. Yeah. So I like to learn more about the stories, but actually I keep the exosome in my refrigerator, not the same day, sometimes 1 week or 2 weeks. And when I to the patient, it still works. I don't know why, but that's why I want to learn from you.
Yeah, that's good. I think I don't have the time. I'm not prepared any proper lecture on that one, but we have carried out quite extensively the quality control of those exosome and the condition media. And we have carried out what is called the microarray data on pre and post. What we are claiming at the moment is max 3 months to 6 months after regulating their shelf life. So we have a technology, I can't dwell on that, what technologies whereby we preserve the half-life of these biomolecules, which can be used, and it has a shelf life. You don't have to refrigerate, you can keep it at room temperature, and that is our technology. I can't tell you what our technology is, but it does work, and I can present some of the slide where we have shown by microarray antibodies assay that the activity of those biomolecules is maintained even after 1 year of preservation on shelf life at room temperature. Yes. Otherwise, the shelf life is not more than an hour and a half. That's it.
Desmond?
Yes, I am very nervous about this, and the first time I come across this is.
But you have a most fertile
Bambang,
brain.
you know?
You have a most fertile brain.
No, I'm learning a lot. I'm just trying to find out. I think this is fascinating. Even though you can say that yes, individual cases and stuff, and unfortunately, in order to convince the larger population to understand this, then we have to have a proper presentation, proper randomized trial, and I think this is very important to keep the credibility of this way of dealing with problem.
Dr. Hanif?
Yes, thank you very much. You listen to me? So I have one question from Dr. Kaldeep. What about the immunogenicity and the allogenic reaction to this? Because we are using exosome from somewhere else. And what are the chances of immunogenic reaction in the patient we are injecting it?
You're asking me the question, right?
Yes.
Well, I mean, that's a good question. As far as the condition media is concerned, the mesenchymal stem cells don't have the histocompatibility antigen, so they don't cross-react. And that is the beauty of using these cells for the patient. They are not rejected, and that's the beauty part of it. Exosome can express some histocompatibility antigen 1 to some extent, but not hugely. So we are in a way, in a very privileged situation to use mesenchymal stem cell or the mesenchymal stem cell derived product, which will not evoke any major immune response in the patient. And also, if you are giving intrathecally, then some of things are even better in terms of immune response.
Thank you very much. After another question.
Yeah.
Sometimes in spinal cord injury, the spinal cord is very much crushed, and there is a little bit gap between the two ends. So how to address in certain cases where there is complete cross-section of the cord? Do you use some sort of bioscaffold for that, or should we use it, or should we use just an exosome in this case?
That's a great idea, but I'm not a spinal cord expert. I'll pass on that question to Bambang, who has been doing quite a bit on spinal cord injury. But your idea of using biomaterial is a very relevant one. And most of the development in stem cell field is happening even in our lab using some of the scaffold. So scaffold made of several different biomaterial are very relevant in bone repair, even in wound healing.
think,
I can talk about.
I think this idea is already in the field. You can use biologically compatible rather
Yes.
than made of any other material. Collagen is a very good scaffold, and
Yeah.
then certain other collagen varieties they are act as a good scaffold, and they can carry your product there, and they can act as a sort of tissue on which the cells can grow and establish the reconnection.
Yeah.
But again, the scaffold business is still in the early phase because they are the different tissues which might create some allergenic or other adverse reaction there. Because I think you have to be very careful in using those materials.
Yeah, but I
Especially
think this is
biological.
very relevant. I think this is very relevant, particularly in bone healing. We are doing some trial with the, what is called a thermosensitive gel.
Hmm.
Now, this is a very interesting gel which is liquid at room temperature and sorry, at 4 degree. And when you apply on the wound, it forms gelation. And we are putting in stem cell in thermosensitive gel to see the viability of these stem cells. Unfortunately, this gel doesn't host these stem cells very well. And we are now going away from using stem cell, but we are using exosome and the condition media in this thermosensitive gel and doing some trial for the wound healing and the bone healing.
We have Professor Nazir, who's a neurosurgeon, and I think he would like to have some comments regarding the exosome use in brain ischemia and valve situations.
I think the result doctor has shown are like magic. You just within 24 hours, patient starts moving, their limbs are working, and I don't think so. This is like medical treatment. It is like a black magic. It must be very effective if it is like that. Then it must be propagated to use in other centers as well.
Thank you. Any comments, Bambang?
Yeah, what we want to do in the second animal study is using the dog and make them in chronic condition of spinal cord injury, complete spinal cord injury, then give this kind of medication. Because using rat, we cannot make the chronic. The rat already died. So I only have the complete spinal cord injury in acute, subacute, and intermediate. So because the histopathology will tell us anything, the truth, before and after.
Thank you. Any further comments by Dr. Kuldeep?
I'm fine. I think I'm excited looking at some of those, the recovery which Dr. Bambang has shown, but I think it has to be proven in terms of publicizing or publishing this data in a good journal so that we can convince people.
Thank you. Thank you.
Thank you very much. Nisa, anything you would like to say?
Yes, thank you very much. We have got a lot of patients of spinal cord injury in our unit, and they are waiting for some miraculous change so that they can start resume their normal routine activities. And we are also very much enthusiastic to see and moving towards a new era so that we can get something better for these patients, for our patients. And I hope we are with you, and we are conducting the different studies, and in future we are doing this job, inshallah. And we will see, and we will get all the record of those patients.
Yes.
Nothing to add on.
Yes.
Nothing to add on.
Nothing. More questions? Kuldeep, you wanted to present something about. You said you will present it at the end. Are you still,
Probably, if you want me, I can show very briefly. Did you disable the participant screen sharing?
no, I don't think so.
Doesn't allow me to share the screen with you because it says the host has logged in. unfortunately, probably
No,
maybe in terms.
then there is some technical issues in sharing at this stage. So I think
Yeah.
we
So there is some. The I think primarily, as I said before, and I agree with the comment from Divok and other, particularly Hanif, that we have to go a long way to understand the mechanism, and understand the mechanism, and then prove uh, with the case control studies. At least have a control studies with them, have a kind of a proper data to publish to convince the people.
Sure.
And I have absolutely no doubt that these stem cell and the stem cell drive product can do miracle, because this is a part of our body. We are trying to exploit the existing system which is performing their role in a repair mechanism within our own body. And if we can exploit that, that is the way to go. COVID-19, I'm look forward tomorrow, because that is where I think uh, probably you are aware of one of the Australian company, Mesoblast, has gone to phase 2, phase 3 clinical trial with the COVID-19 using their product, which is very similar to Endizone, which we have prepared, and we are trying to find partner to do some clinical trial. I will not go over the board by claiming that we have done the treatment. We haven't done the treatment. We are in the pilot studies, and we want to get the confidence that what we are saying and looking at the mechanism, and then we follow through, and then exactly do phase 1, phase 2, phase 3 clinical trial before we convince other people. Because I come from an academic background, I have to convince myself first before I can make a claim that it helps. But it helps in isolated patient. That might be a placebo effect. When the patient come to the doctor, even doctor touches that patient, patient get better. So the placebo effect can be very, very strong sometimes. You never know. But we need to nullify, or at least look at whether it's a placebo effect, or it's actual effect of the cell or the product going into the patient and doing the wonder, as we have seen in some of your slide. But there is a huge potential in this field. We should continue to endeavor and collaborate, and then generate some data to convince people that this is how it happens.
Sure. Thank you. Thank you for your comments, Kuldeep. I thank you, Bang Bang, for very encouraging and very exciting presentation, and some thoughts behind the new direction which we are learning every day. And I thank you, Des, for your very thought-provoking comments. And we always look forward for your very philosophical approach to the whole issue, whatever issue we present to you. And Hanif, you have been very instrumental in, getting on with this idea. And I think hopefully with the collaboration of Bang Bang, Kuldeep, and other colleagues, we will, inshallah, move forward and help these patients in coming back to their feet. And hopefully, I think I'm looking forward in a very excited way and a very positive way to get along with and help those people. And I thank you very much, all the participants and all the people who are connected and listening to this. If anybody from the audience, they have any question, they can field this question to either to speak themselves, or they can put up through me.
Yeah. Oh, that's a good question. I think I got some question from Dr. Michael. There are different ways to isolate exosome, but one of the issue I always overemphasize on the quality control. You have to make doubly sure that what you are saying you are actually doing. So isolation of exosome, people use ultracentrifuge to purify those exosomes. In our lab, we use ISORD column. Now, these ISORD columns are very expensive column. Why we use them? Because they are GMP compliant. So when we prepare exosome using ISORD column, you get a GMP compliant product which we can try in these patients. It's very easy, most convenient, but more expensive. But if you want to bulk produce those exosome, I think you have to go ultracentrifuge and produce a mass production of those exosomes. We are not at that stage to mass produce, but I will prefer to concentrate on looking at the mechanism using GMP compliant exosome coming out with ISOD columnar technology. Does that answer your question, Michael? Yeah, that was a question for Michael anyway.
Okay. Thank you very much, Kuldeep. Thank you very much, Bang Bang. Thank you very much, Des. And thank you, Hanif.
Thank you very much, Dr. Tariq, for taking that initiative. That was great.
I think
And also
this is the first of the few webinars we will meet and conduct these regularly so that we can have an exchange, upgrade ourselves, and improve our understanding and of all these issues. I think it's a very exciting field. It's a way forward, to be honest.
So, sure. What time is the meeting tomorrow?
1 o'clock, same time.
Same time.
Yeah, okay.
All right. Okay. All right. You will send another invite or the same
tomorrow,
invite?
as well, Hanif sir, तुसी भी आ जाना. ठीक है?
I will.
So you will.
Tomorrow, COVID-19 and exosomes. Spine surgery and exosomes.
One question to Hanif.
Yeah.
Yes.
Do you miss that duck tour in Boston? Have you done a duck tour? That's the most exciting, isn't it?
Yes.
You take a duck tour and you become amphibious, goes into the lake, and it's a beautiful view over there. Billionaires on one side, and it's an interesting experience in Boston.
Thank you, gentlemen. Thank
All
you.
right.
Thank you all the participants who listened to and participated and asked questions. And I thank individually all of you to participate, and hopefully we will conduct these similar meetings in future.
Thank you so much. Appreciate it.
Thank you.